we are trying to understand the regeneration step involved in the C3 pathway. We know that in the regeneration step, in the 3-carbon products, which are glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, must be recycled in order to make ribulose 1,5-bisphosphate. So here we have glyceraldehyde 3-phosphate, glyceraldehyde 3-phosphate plus dihydroxyacetone phosphate, CH2OH. This is the dihydroxyacetone phosphate. This must be converted to ribulose 1,5-bisphosphate. Let us understand that these were all laborious experiments that were carried out in the 1940s. So Benson explains in his work that he was using C11, carbon 11, initially for radio labeling. C11 has a problem. The problem is its half-life is only about 21 minutes. So therefore it becomes extremely difficult to identify the products which are being formed. Later on he switched over to C14. So we know that the half-life of C14 is roughly about 5,730 years. So therefore, and it was very easy to get to C14 in those days in comparison to the C11 isotope. So the studies were carried out by using C14. Now, there are several enzymes, they will be repeatedly used in order to get ribulose 1,5-bisphosphate. In the first step is a kind of a condensation reaction, and this is known as an aldol condensation reaction. An aldol condensation is when a ketose and aldose join together in order to form a larger aldose. So, a larger ketose. That is an aldol condensation reaction. So what happens in this step is this ketose is getting added to this aldose, and you are going to get a 6-carbon product. The 6-carbon product is this is the 6-carbon product, and this 6-carbon product, we know that this is nothing but fructose 1,6-bisphosphate. So, and this is catalyzed by an enzyme known as trans-aldolase. We will write short form TA, trans-aldolase enzyme. So therefore we are getting a 6-carbon product. This is the first step in the regeneration. In the next step, what happens is this fructose 1,6-bisphosphate is acted upon by an enzyme known as fructose 1,6-bisphosphatase. That is hydrolyzing one of the inorganic phosphate from the first position, and the end product is going to be fructose 6-phosphate. So once we have fructose 6-phosphate, we'll write the structure. Once we have fructose 6-phosphate, here is CH2OH. This is fructose 6-phosphate. Now, if the fructose 6-phosphate, a 6-carbon sugar, will combine with a 3-carbon sugar. And if the 3-carbon sugar, now this is a ketose, the next 3-carbon sugar should be an aldose. Now, this is going to be catalyzed by an enzyme known as transketolase. Transketolases are enzymes which will transfer 2 carbon units to the next molecule here. And the 3 carbon, therefore one has to be a ketose, other one has to be an aldose. What we will do, the 3 carbon aldose is glyceraldehyde 3-phosphate. So if the 2 carbon is transferred to glyceraldehyde 3-phosphate, so this becomes a 5 carbon ketose. And the 5 carbon ketose is this is the 5 carbon ketose, and that is cellulose 5-phosphate. So once you have cellulose 5-phosphate, in the remaining 1, 2, 3, 4 carbon is an aldose, and we will write it here. The 4 carbon aldose is this is erythrose 4-phosphate. Erythrose 4-phosphate. Since there is no space, I am just writing a short erythrose 4-phosphate. We are getting 1 ketose and 1 aldose. In other words, C6 plus C3 transketolase enzyme that will result into C5 plus C4. This is the next step. After this, if the next step is going to be catalyzed by 1 aldose and another ketose will react. Which are the aldoses and which are the ketoses? So remember that we have already created 3 carbon, 4 carbon, 5 carbon and 6 carbon sugars. If the next reaction is going to be between a C4 plus a C3, that will result into a C7. So this is the next reaction. And which is the C4? If the C4 is going to be, we have only one C4, that is an aldose, which is erythrose 4-phosphate. So erythrose 4-phosphate plus, since it is an aldose, the next molecule should be a ketose. If the 3 carbon ketose that we have is dihydroxyacetone phosphate, and they will join together with the help of an enzyme transaldolase. Remember, transaldolase will transfer a 3 carbon unit. It will transfer a 3 carbon unit and they will join together in order to form a 7 carbon unit, and the 7 carbon unit is this. This is the 7 carbon unit. The 7 carbon unit is pseudoheptulose 1,7-bisphosphate. There is an interesting story that Benson narrates about the discovery of pseudoheptulose. How he isolated pseudoheptulose by using the 2-dimensional paper chromatography. When he identified, he was excited to share the news with his friends, and he went around looking for somebody to share the excitation that he has identified a new sugar molecule. In the next step, once we have pseudoheptulose 1,7-bisphosphate, is to convert this into pseudoheptulose 7-phosphate, which means this phosphate must be removed. As we know, it will be catalyzed by pseudoheptulose 1,7-bisphosphatase. As we have seen in the earlier case, it is going to be a hydrolytic reaction. Inorganic phosphate is removed. You will end up by getting pseudoheptulose 7-phosphate. You remove the phosphate group from there, that will be the 7 carbon pseudoheptulose 7-phosphate. Remember, this is an irreversible reaction. In the regeneration, we have already encountered 2 irreversible reactions. So now we have a 7 carbon compound. Let us see what happens next. In the next step is our target is to make ribulose 1,5-bisphosphate. Now we have 3 carbon, 4 carbon, 5 carbon, 6 carbon and 7 carbon sugars. So our next step is C7, that is a ketose, combines with the C3 So totally there are 10 carbon atoms. You will be able to make C5 plus C5. This is going to be the next step. C7 is going to be pseudoheptulose 7-phosphate, and the 3 carbon sugar is going to be, therefore this is a ketose, the next one has to be an aldose. So this is going to be glyceraldehyde 3-phosphate, and they will split into, so this is going to be 2 carbons are removed, that is a ketose will be removed, so therefore it is a trans ketonase reaction. You will get now 2 products. The 2 products are, one is this is cellulose 5-phosphate, and the remaining 5 carbons of pseudoheptulose will be this is ribose 5-phosphate. So by using this series of enzymes like transketolase and transaldolase, finally we have got 2 5-carbon products, cellulose 5-phosphate and ribose 5-phosphate. Our next step is to synthesize ribulose, which is a ketose, from these 2 products. How do we do that? That is going to be the last step. So in this, what we do is, in the structure of ribulose 5-phosphate is this. This is the structure of ribulose 5-phosphate. We are left with the 2 products. The 2 products are cellulose 5-phosphate and ribose 5-phosphate. These are the 2 products that we have at the end of it. Now, cellulose 5-phosphate is an epimer of ribulose 5-phosphate. So therefore, there is an enzyme known as ribulose 5-phosphate epimerase. So this enzyme will convert cellulose 5-phosphate into ribulose 5-phosphate. Simple reaction. Similarly, ribulose 5-phosphate and ribose 5-phosphate and ribulose 5-phosphates are isomers. So therefore, ribose 5-phosphate isomerase will convert ribose 5-phosphate into ribulose 5-phosphate. So this is how the production of ribulose 5-phosphate is taken care of. We know that this is not what we wanted. We wanted ribulose 1,5-bisphosphate. How do we convert ribulose 5-phosphate into ribulose 1,5-bisphosphate? This is carried out by a simple kinase enzyme. An ATP is added in the 3rd irreversible reaction in the C3 pathway. ATP hydrolyzes to ADP. Inorganic phosphate is attached here. You will get ribulose 1,5-bisphosphate. Name of the enzyme is phosphoribulokinase. So by using these enzymes, we are converting the 3-carbon unit into the 5-carbon ribulose 1,5-bisphosphate. So this is how the regeneration takes place. Let us summarize the whole pathway in the form of a chart. This is in a way the summary of the whole C3 pathway. So we began from here, ribulose 1,5-bisphosphate, carbon fixation has happened, and then the reduction process has happened, and this is the regeneration process. Finally, we have reached ribulose 1,5-bisphosphate. In this, there are 3 irreversible reactions. This is an irreversible reaction. This is an irreversible reaction. Here, this is an irreversible reaction, and this is an irreversible reaction. So therefore, there are 4 irreversible reactions which are used for regulating the C3 pathway. After having understood the C3 pathway, let us look at the overall stoichiometry of this pathway. This is the overall stoichiometry of the C3 pathway. 6 rounds of carbon fixation is required, 6 rounds of Calvin cycle. That will require about, every cycle requires about 12 ATP molecules and 12 NADPH. Now, another additional 6 ATP molecules are required in order to convert ribulose 5-phosphate into ribulose 1,5-bisphosphate. So therefore, there is an additional 6 ATP added, so which makes it 18 ATP required for fixing 6 molecules of carbon dioxide. This is the energy requirement for C3 pathway.